Welcome everyone to our November Grand Rounds. We have with us Dr. Munevver Dogramaci, who is a scientist at Stanford Imagenetics and assistant professor at USD. Dr. Dogramaci earned her Master in Science and PhD in Turkey and is specialized in molecular biology, genetics, and genomics. We're going to have also Dr. Ellithi and Dr. Shahid, who are second-year residents. And Dr. Ellithi is internal medicine resident. He was a research fellow in Philadelphia before starting his IM residency with us at USD. Dr. Shahid, she's also internal medicine resident. She is interested in oncology, and she's presently doing research in targeted therapy and cholangiocarcinoma, which is awesome. So for those of you who joined later, we have some evaluations form for Dr. Dogramaci. Please grab one and fill them after the lecture. Leave them on the table, and if you can put on the top that your role. Don't put your name, just if you are a student resident or fellow or pharmacist, attending, whatever your role is. Okay, so please, Dr. Shahid.
Thank you, Dr. Lupu. I am Mahum Shahid. I'm a second-year internal medicine resident, and today we'll be presenting transcriptome profiling and its clinical utility. We have no disclosures to make. I'll be starting the session with a case report that would highlight how this technique can actually impact our clinical decision making in the future. This will be followed by an overview by Dr. Dogramaci, and in the end, Dr. Ellithi would come up and wrap up the whole session. So this case report was taken from Journal of Clinical Oncology, Precision Oncology. It's a pediatric case. An 8-year-old male patient presented with 2-week history of nausea, vomiting, and lethargy. They did some basic lab work, and during the brain scan, they found a tumor on the ventral aspect of the left side of the tentorium, and it was pressing on the cerebellum as well. They did pathology and immunohistochemistry for this tumor, and they found it to be made of epithelioid and spindle tumor cells. They did some immunohistostaining as well, and the final diagnosis was that this was a poorly differentiated sarcoma. So they went ahead with traditional therapy for it. They gave the patient 6 cycles of chemotherapy. It was followed by autologous stem cell transplant. And in the end, the patient was given some focal radiation to the location of the original tumor. So the patient achieved remission for 2 years. And after 2 years, the patient had recurrence of that tumor. And this time, the tumor recurred in both lungs. So now they had to change their treatment strategy. This patient was inducted in personalized oncogenomic study for whole genome sequencing and transcriptome profiling by the way of RNA sequencing. Biopsy material from a lung metastasis was characterized by whole genome sequencing and RNA sequencing, and peripheral blood was characterized only by whole genome sequencing. So without going into the details of what whole genome sequencing is or what RNA sequencing is, I'll just touch up on the results. So whole genome sequencing actually showed somatic variants of unclear therapeutic significance. There were 3 of them, and no germline single nucleotide variants with established cancer relevance were detected. Mm-hmm.
You did WGS on tumor, or this was peripheral blood?
So whole genome So in this case, whole genome sequencing was done on the metastatic side of lungs, like the lung metastasis and peripheral blood both. And RNA sequencing only on the metastatic lesion that was found in the lung. So the results of the RNA sequencing showed 1 gene fusion. So there was gene fusion of EWSR1 and ATF1 gene. What do these genes actually do? So EWSR1, this gene encodes a multifunctional protein involved in various cellular processes, including gene expression, cell signaling, and RNA processing and transport. chromosomal translocations between this gene and various genes encoding transcription factors result in the production of chimeric proteins that are involved in tumorigenesis. Now the other gene, ATF1, the phosphorylation of this gene enhances its trans activation and transcriptional activities and enhances cell transformation. Fusion of this gene and FUS on chromosome 16 or EWSR1 on chromosome 22, like we see in our patient, induced by translocation generates chimeric proteins in angiomatoid fibrous histiocytoma and clear cell sarcoma. So both of these genes pretty much promote tumorigenesis, if you have to summarize this. Now, RNA sequencing also showed high expression of downstream genes interleukin-6 and JAK1. So the clinicians in the audience might already have an idea what are JAKs. They are tyrosine kinases that engage with cytokine receptors and mediate tyrosine phosphorylation of their associated receptors and recruited proteins, including STATs. So we have heard this name before in cancer as well. Interleukin-6 mediated activation of STAT3 is a principal pathway implicated in promoting tumorigenesis. And this pathway was detected in our patient. So I do feel, being clinicians, we are sometimes spoiled because we get that one-page report and we don't exactly know what happened in the whole process, how we got that one drug to treat the patient. So I'll just very briefly touch up on how RNA sequencing can give us such meaningful data, because what I just talked about did give us a very good idea what was happening with the patient's tumor. So RNA sequencing derived tumor gene expression profile of patient 1 was compared with similarly derived profiles of more than 10,000 samples that represented 38 pediatric and adult tumor types. And this was studied by the Cancer Genome Atlas and therapeutically actionable research to generate effective treatment. When the pathologist looked at the sample of the tumor, 76% of the sample had tumor cells. So it was a good representation of what the tumor was basically composed of. The tumor gene expression for patient 1 resembled lung cancers, which was the site of metastasis for this patient. The sarcoma of patient 1 clustered with 354 lung adenocancer tumors of the terminal respiratory unit, and the subtype was proximal inflammatory subtype, which was associated with the activation of receptor tyrosine kinases. And I am going to explain this in detail in the future as well. So to define the transcriptional programs that drove placement of the patient's tumor with lung cancers, 2 analyses were done. First, gene set enrichment analysis with genes differentially expressed between lung cell adenocancer cluster that contained the tumor of patient 1 and the remaining samples in the compendium. And the second analysis was the comparison of the cluster for patient 1 with the remaining lung adenocancer samples. Both analyses revealed the overexpression of the members of the interleukin-6 JAK-STAT pathway. Outlier statistics were utilized to explicitly subtract the effect of the lung cell expression because the tumor cell was found in the lung in the first place, so they had to subtract that effect from their data. 787 genes, including druggable targets, JAK1, ALK, and TRK1, and CCND1 emerged as overexpression outliers in all analyses. So what I just talked about, let's visualize everything. The colorful picture over there is a tumor map. And I will explain it to you like how I explained it to myself, because I'm a resident myself and I don't have expertise in that. Each hexagon on that tumor map is a tumor cell. The color represents the tumor type. So the difference of color is based on what kind of tumor it is. A projection of the entire tumor cohort in 2 dimensions according to tumor map method was done over here. Individual tumors are represented by hexagons, like I just said, and color tumors by the tumor type. The tumor in patient 1 is shown in green within the lung tumors. So the lung tumors are actually that orange cluster on the bottom right of this map, and we will zoom into this as well. So lung adenocancer actually formed 4 clusters on the tumor map, and these 4 clusters are actually seen on the slide as well. The patient's tumor was found on this cluster. And how did they find that the patient's tumor was found in this cluster? They studied the gene expression of this tumor, what kinds of proteins were produced by that tumor, and they felt the tumor of the patient was very close to how lung adenocancers produce their own proteins. So that's how the patient's tumor was placed into that cluster. Now, I did discuss about this molecular subtype of our patient's tumor, and it was proximal inflammatory type. So now we are further zooming into that orange cluster we saw before. It's the same cluster. Now the different colors show different types of molecular subtypes of the tumors that this cluster is composed of. And our patient's tumor was actually found over here. So just summarizing everything, why was it important to place our patient's tumor in that cluster? Because we want to know how that kind of tumor would act based on the similarity it has in morphology and functioning to other tumor cells. So I think it's reasonable to believe that if our patient's tumor was found in the lung adenocancer cluster, it would act like it because it's producing similar sort of proteins. Now, this kind of tumors, the lung adenocancers, they have overexpression of receptor tyrosine kinases. So we assume that the patient did have overactivation of receptor tyrosine kinases. So I think this diagram will be more helpful in understanding this. We felt that our patient had two mechanisms that caused overexpression of that JAK1 STAT pathway. The fusion protein EWSR1 and ATF1 had downstream interleukin-6, which can actually trigger JAK and can initiate that pathway. Now, receptor tyrosine kinases, which are generally found in those lung adenocancers, they have overactivation of these RTKs, and they can also induce overexpression of JAK1. So this patient actually had two reasons to have overexpression of interleukin-6, JAK, and STAT3 overexpression. Now, just based off this diagram, if we have to target something that can stop that overexpression of JAK1 STAT pathway, I think it would be reasonable to target this JAK1. So this is what they did with this patient. Ruxolitinib is a drug which is a JAK1 and JAK2 inhibitor, and this drug was given to our pediatric patient. They did not have very significant data in terms of dosing of this drug in pediatric patients, but they somehow read how this drug is studied in adults, and they went with the same sort of dosing for this patient. And this patient was started on this drug. After 1 week of initiation of this drug, patient had dramatic improvement in his energy level, his nausea resolved, his previously growing lung nodules stabilized, and there was improvement in his landscape performance score. So landscape performance score is something very similar to ECOG scale, but it's for pediatric patients who are less than 16 years of age. 10 will show a patient who's very disabled and who would not even involve in passive play, and 100 would be a normal child with normal activity level. So this patient who had a landscape performance score of 60, he actually went from 60 to 90 to 100. So that was a overly dramatic improvement. So on follow-up, and I'm not sure after how long, but they did some routine imaging as part of the follow-up for this patient. And this patient had enlargement in one lung lesion, although other lung lesions remained stable. Now, ruxolitinib was discontinued, and focal palliative radiation was administered to one lesion in the left lower lobe for pain control. Within 2 months of stoppage of this drug, the symptoms of nausea, extreme fatigue, and weight loss returned, and the lung lesions progressed. Now, the family actually requested that the drug should be restarted for quality of life, and the patient again showed dramatic improvement in clinical status and an unexpected prolonged period of stable disease until dose reduction had to be done because of myelosuppression. After the dose reduction, a rapid progression of the lung lesions resulted in death of the patient in 23 months after the original relapse. Now I would invite Dr. Dogramaci. Sure. Mm-hmm.
So I want to make sure I understand this right. So their workup with RNA-seq showed that this is an adeno-primary in the lung and not a metastatic lesion from the sarcoma, or it was a metastatic lesion from the CNS sarcoma.
So it was a lesion from sarcoma, but in terms of gene expression, it resembled lung adenocancers. Yeah.
Maybe even the CNS lesion doesn't primarily use metastatic. But this is a sarcoma, and it's all the same cancer. You just have a recurrence here. You did RNA-seq on the lung mets.
Yes.
And then you predicted that the lung mets were going to behave like an adeno, and you directed targeted treatment at them.
That's
right?
correct, yeah.
All right. For us to understand how they made that decision, show me the cluster plot. Yeah, one more back up there. Yeah, and I'm not roughing you up. This is super cool. I want to understand. That's not histology, right? This is a cluster plot where, based on thousands of RNA-seq done on thousands of tumors, these are different combinations in a two-dimensional array.
Each dot represents the patient's tumor molecular data in here.
Gotcha.
So,
Thousands of tumors, each dot is a patient.
So is that the, oh, sorry, the UCC Trios project? Is that the
Yes,
okay,
yes.
okay.
And so where you fall on this map reflects the combination of genes that your tumor has upregulated.
yes, so
Okay.
it defines the similarity and differences between the different tumors.
Gotcha. And your patient fell within a certain cluster that tends to have a pattern of gene upregulation, which includes JAK.
Mm-hmm.
Gotcha.
Well, so I had a question about this as well. You're saying that the tumor that you biopsied has more of a lung cancer pattern, expression pattern.
In terms of expression, yes.
Is that because it was just located in the lung tissue, and you were also biopsying lung tissue as well? I think you said, what,
36% of the tumor, sample was composed of tumor cells. And you are right, just based on the fact that this tumor cell was found in the lung, it would affect its expression of proteins. But then they apply something called outlier statistics to subtract that effect of the fact that this tumor sample was taken from the lung itself. So once they subtract that effect of it being originated from from the lung itself, they would be able to sort of comment on how the tumor is acting in terms of its gene expression.
So lung or not, it still fell into the pattern where it is rich with JAK expression.
right? Yeah, so the JAK expression, I do believe would probably be coming from the tumor itself, right?
Yeah.
My question was, is it giving a lung pattern because you're looking at, whatever percentage was normal tissue to lung tissue, right? Or lung, the tumor tissue.
Either way, it got us to the target, though.
That's true.
The JAK target.
Yeah.
Thanks for the nice presentation, Dr. Shahid. And I would like to thank Dr. Rupe and Dr. Wilkie for inviting us today to give the talk. So in this section, I will mainly focus on transcriptome profiling, but before we get there, I'd like to go over some basic information so we are all on the same page. So the human genome refers to the complete set of nucleic acid sequences encoded as DNA within the 23 chromosome pairs in cell nuclei and in a small DNA molecule in mitochondria. It consists of about 3 billion base pairs and it contains about 21,000 protein-coding genes. More than 99% of the human genome is the same in all people, and differences in less than 1% of our genome accounts for the diversity of humans across the globe. An individual's genetic code contains millions of differences when compared to the reference human genome sequence, and these differences, referred to as variants, are sometimes also called mutations. Most variants are considered regular and common changes and generally related to the traits of individuals such as eye color, skin color, hair texture, but a small number of variants are considered disease-causing. Genetic variants may be benign and have no impact, or may be pathogenic and causative of disease. When it is unclear whether a variant has an impact, it is referred to as a variant of uncertain significance. variation in the genome across species can be measured via sequencing and elucidates recombinations, deletions, duplications, copy number variations, or single variations. So although DNA controls the working of the cell, it is trapped inside the nucleus, so it must deliver its genetic code to the rest of the cell. In order to deliver all of its instruction, DNA relies on RNA. So RNA's job is to help DNA make proteins. And protein synthesis requires transcription and translation processes. During transcription, messenger RNA is transcribed from DNA's code and then transported into cytoplasm for protein synthesis. The identity of a cell is specified by the collection of genes actively expressed within that cell. So gene expression patterns vary widely in complexity. Some are straightforward and static, which is expressed constitutively in all cells at all times in life and referred to as housekeeping genes. But some are extraordinarily intricate and difficult to predict, with expression fluctuating from cell to cell, from minute to minute. So spatial and temporal gene expression indicates the activation of genes within specific tissues of an organism that is spatial at specific times during development or also during diurnal cycles, and that is temporal. And both spatial and temporal gene expression plays a key role in generating the diversity of cell types. So it's well known that identity of all organisms is embedded in the genome and then differentially functionalized based on the developmental and environmental cues. This functionalization of the genetic code progresses through transcription, as just mentioned, it's synthesis of messenger RNA, translation, synthesis of proteins, and metabolism, regulation of metabolites. And the graph here illustrates the omics organization. And each of these processes operates at different temporal scale. So acquiring high-throughput data via omics platforms and openly available bioinformatics toolsets and resources for data analysis and interpretation opened a new era in biological sciences. By gathering high-throughput data from these analytical platforms, such as next-generation sequencing, one hopes to postulate meaningful hypotheses, some of them to address complex biological questions such as disease mechanisms. Integrated omics may help elucidate the spatial and/or temporal dynamics of these biological complexities. Many institutions have already integrated DNA or RNA-based tests as preemptive or diagnostic tests. Also, multi-omics approaches to have a better understanding of molecular mechanisms associated with diseases to guide diagnosis and therapy to achieve optimal health outcomes. Again, our focus is transcriptome profiling today, but I would like to go over DNA-based analysis just briefly. So DNA-based analysis usually refers to one of these four: genotyping, gene panels, whole genome sequencing, and whole exome sequencing. Genotyping is performed to determine the presence or absence of specific gene variants in certain locations in the genome where variations often occur. Focuses on specific bases in the genome known to vary from person to person. And good example for this is Sanford chip. And there's plenty of educational material and information on this on Imaginetics website if you would like to learn more about it.
How many SNPs on the Sanford chip?
Over 700,000. I believe 740,000.
Out of 3 billion nucleotides.
Yes. So gene panels are mainly utilized for patients with certain diseases. For example, cancer gene panels that include more than 100 genes which are currently being evaluated for efficacy in determining targeted therapies. Whole genome sequencing is the process of reading every single letter in an individual's genetic code. And it covers everything: exons, the regions coding for protein, introns and regulatory non-coding DNA, unique non-coding DNA, and also repetitive DNA.
So that's all 3 billion, and you would get the 700,000 nested within that 3 billion.
Yes. Well, 3 billion is in the haploid genome, and diploid genome has about 6 billion. So, whole exome sequencing focuses exclusively on the protein coding regions of the genome. That's about 1 to 1.5% of the whole genome. So it is based on selecting and capturing only the subset of the DNA that encodes for proteins, and sequencing the exonic DNA to identify genetic variants that alter protein sequences. So both of these DNA-based sequencing approach have already been implemented as a clinical test by many institutions. I must note here, though, whole exome sequencing is able to identify those variants found in the coding regions of genes, but it is not able to identify the non-coding variants associated with the disease because you don't sequence that part. Whole genome sequencing has the potential to observe variants missed by whole exome sequencing. However, the increase in the diagnostic rate attributed to whole genome sequencing has been nominal so far, because variation in non-coding region is less well understood than variation in the coding regions, making it more difficult to predict which variants might be relevant to a trait of interest because of whole genome sequencing results bring a lot of variants of uncertain significance. Both of these DNA-based sequencing approaches also exclude information about interaction with genome with the environment. It's well known that nearly all diseases result from a complex interaction between an individual's genetic makeup and the environmental factors he or she is exposed to, such as diet, exercise, smoking, alcohol consumption, exposure to UV light, or chemical exposure, or air pollutants. And differences in genetic makeup may cause people to respond differently when exposed to the same environmental conditions. Transcriptome profiling has the potential to capture this dynamic nature of genetic environment interaction. So transcriptome is the complete set of RNA transcripts that are expressed by the genome under specific circumstances or in a specific cell. And transcriptome profiling provides a snapshot of continuously changing cellular transcriptome, and it is essential for discovering molecular mechanisms associated with diseases. Different technologies have been developed to quantify transcriptome, including hybridization-based microarrays, and recently this is replaced almost completely by sequence-based approaches. And the next generation sequencing technologies provide reliable method for quantifying transcriptome profiles, and it's termed as RNA-seq. So RNA-seq studies are diverse in their goals, therefore sample preparation, sequencing, and analysis pipelines vary based on those objectives. And we are not going to get into any technical details today. Just I would like to highlight the key barrier of transcriptome-based analysis in general is RNA quality and the need for source material with disease representative expression patterns. Also, there are several other factors to be considered that impact the results, such as selection of reagents. For example, it can impact the RNA to be sequenced. RNA exists in several different single-stranded structures, most of which are directly or indirectly involved in protein synthesis or its regulation. And the graph here shows different classes of RNA involved in protein synthesis, or post-transcriptional modification or regulation of gene expression. Usually, when RNA is extracted, messenger RNA accounts for only 1 to 5% of the total cellular RNA. The majority of RNA molecules are ribosomal RNAs. Therefore, RNA purification is necessary to enrich specific classes of RNA in the sample to be sequenced. The goal here is, by enrichment, not to sequence uninteresting or uninformative RNA, because role of both ribosomal and transfer RNA well studied, and they are involved in protein synthesis. So, common methods used for RNA enrichment include poly-A selection to enrich messenger RNA by taking advantage of its poly-A tail and using a right chemistry to capture, and RNA depletion to get rid of ribosomal RNA and capture some regulatory RNAs as well as messenger RNA. So poly-A selection is an efficient method for capturing messenger RNA, and it may be sufficient if the goal of experiment is differential gene expression. But ribosomal RNA depletion may provide a deeper understanding of transcriptome activity and facilitate novel discoveries. So poly-A selection is like if you are interested in catching certain fish, using a rod and using a right bait to catch that fish. But if you want to know what kind of fish is available in the water, you probably using a net is a better choice. RNA goes through several steps before it can be sequenced using current next generation excuse me, sequencing instruments, because current instruments do not directly sequence RNA, they sequence fragments of DNA. So RNA must be converted to double-stranded complementary DNA libraries. cDNA library refers to collection of cDNA fragments which are flanked by specific constant sequences called adapters that are necessary for sequencing. So library preparation basically involves fragmentation of RNA, synthesis of cDNA, construction of cDNA libraries, and amplification prior to sequencing, and using one of these next generation sequencing platforms to generate millions of reads. And here, after that point, those short sequence reads go through the data analysis pipeline. And in brief, data analysis involved initial filtering of sequencing reads, removal of adapters, adapter sequences, aligning the short sequence reads to reference sequences, annotating the transcripts, quantifying the number of reads per transcript, followed by various downstream data analysis.
And then you get the cluster plots that you were showing us.
I'm going to get there in a minute. Okay. So downstream data analysis may include one or many of these. So these are commonly used ones: differential gene expression, principal component analysis, gene set enrichment analysis, alternative splicing detection, gene fusion detection, tumor map analysis. Also, Dr. Wilkie just was asking me if I was going to talk about heat map. I didn't include that one here. Among many goals of RNA-seq study, identifying differential expressed genes between usually two conditions is probably the most common one. Comparison of transcriptomes allows identification of genes that are differentially expressed in distinct cell populations or in response to different treatments. For example, differential expressed genes between normal and tumor samples help to study tumorigenesis and have been routinely applied to identify diagnostic, prognostic, and therapeutic biomarkers for many cancers. To understand normal cell development and disease mechanisms, researchers frequently investigate differential expression during development in specific tissues or in response to varying conditions as well. Sometimes differential gene expression analysis may identify several hundreds genes, or in some cases a few thousand differentially expressed genes. When a large number of genes pass a significant threshold for this analysis, differential expressed analysis, there may be no obvious way to interpret and identify the relevant biological process that those genes represent. So at that point, complementary tools like gene set enrichment analysis may be helpful. So gene set enrichment analysis is a method that determines whether a predefined set of genes show statistical significance between two biological conditions, for example, tumor versus normal. It focuses on coordinated differential expression of gene sets and produces results that can more easily be interpreted. For this analysis, a molecular signature database was developed by Broad Institute and University of California, San Diego many years ago. And the database contains about 18,000 gene sets divided into 8 major collections, and each collection has several sub-collections. For example, the hallmark gene set is one of those 8 collections and contains contains 50 subsets.
These are gene networks that can help the non-regulated together.
These are gene sets, yes, in certain pathways, and you may be, yes, and you may be familiar with some of these. I'm gonna, for example, reactive oxygen species pathway, MYC target pathway, P53 pathway, mTOR signaling, NOS signaling, TGF-beta signaling, TNFA signaling pathways. And once this analysis identifies certain gene sets, you can focus on certain pathways and narrow down the genes of interest we study. RNA-seq studies, RNA-seq data provides more information than just a transcript abundance and allow, for example, detection of alternative splicing patterns. I would like to remind what RNA splicing here is. So it is a post-transcriptional process in which a newly made precursor messenger RNA transcript is transformed into mature messenger RNA. And 2 forms of splicing have been defined: constitutive splicing and alternative splicing. Constitutive splicing is basically the process of removing introns from the pre-mRNA and joining the exons together to form a mature mRNA. And alternative splicing is the process where exons can be included or excluded in different combinations. The graph here illustrates precursor, newly transcribed precursor RNA, including 5 exons shown in color boxes and intron as a line. So during constitutive splicing, all introns are removed and exons are merged to form the messenger RNA. In alternative splicing scenario, the first one excludes the exon 3, and the second one excludes the 4th exon. So this also shows that 3 different isoforms of messenger RNA can be generated from precursor RNA. And this is just a simplified version of alternative splicing patterns. And in real life, it may be a little more complicated than that. And basic alternative splicing patterns include exon skipping, alternative 5 or 3 splice site selection, mutually exclusive exon, that is one of the exons are skipped but not both, intron retention, sometimes introns are not removed, and alternative first or last exon selection. And there are more complicated versions of alternative splicing patterns. So this alternative splicing is a natural process, and it is tightly regulated, by which a number of gene products for a given gene can be greatly expanded. So although, as I mentioned earlier, the human genome has about 21,000 protein coding genes, the unique messenger RNA isoforms generated can be more than 10 times that number. And almost all of the multi-exon genes go through alternative splicing process. And in some extreme cases, several hundreds isoforms can be generated from a single gene. So alternative splicing, again, is a natural process and regulated by an extensive protein-RNA interaction network. But studies indicate genomic variants in splicing regulatory sequences can disrupt splicing and cause aberrant splicing patterns, and can result in diseases by modifying important protein functions.
Sure, one of our important pharmacogenes that you present for CHIP is an example of that. CYP385 star 3 pack.
Which one?
CYP385 star 3. Star 3 is a genome variant that causes a splice variant.
Good to know.
Yeah.
Studies highlight the involvement of alternatively spliced isoforms in various diseases, including neurodegenerative disorders, cancer, immune diseases, cardiovascular diseases, and also metabolic conditions. Therefore, better understanding of the disease mechanisms and development of clinical applications may require the integration and interpretation of alternative splicing patterns. And many computational tools have been developed for detecting these alternative splicing patterns from RNA-seq data. Another clinically relevant information can be gleaned from RNA-seq data is gene fusion detection. Chromosomal rearrangements that bring two or more genes together can form a gene fusion, as it was in the case presented by Dr. Shahid earlier. So gene fusion detection is particularly significant in tumors, as about 20% of all human tumors have been shown to carry translocations and gene fusions. Therefore, the rapid and accurate identification of gene fusions can characterize and stratify cancer diagnosis, inform subsequent therapeutic treatment, predict prognosis, patient survival, and treatment results. Traditionally, fluorescent in situ hybridization and RT-PCR methods have been used for gene fusion detection. These methods are highly sensitive, but typically only test for the presence of a single fusion gene at a time, and may not be able to identify complex structural rearrangements. RNA-seq can address these limitations by providing a wide surveillance of transcribed gene fusions with nucleotide level resolution. Again, numerous bioinformatics tools exist for fusion detection. Tumor map analysis is fairly new compared to the other analysis methods I just mentioned. This is developed by University of California, Santa Cruz Genomics Institute. It's a public portal to assist in exploration and statistical interrogation of complex omics data. It is a multi-dimensional scaling method using a spatial correlation analysis where samples are arranged on a grid based on their similarity to one another so that nearby samples are like one another. So I read this paper several times, and this is all I can say. And I passed the paper to Adam yesterday, so if you have more details, I will just refer to him. Okay, so this portal allows tumor transcriptome data to be uploaded and plots individual tumors as dots that cluster together, forming a topology of genomic similarity. It analyzes transcriptome data in the context of large data sets to identify situations where an available drug is predicted to work for a given patient's tumor. The goal is to learn how a tumor differs and what it has in common with other people's tumors on a molecular level. So this all started with Kelvin, according to UCSC Trials Child Cancer Initiative website. He was diagnosed with cancer when he was only 8 and helped inspire the Trials Child Cancer Initiative. He is also the patient one in the case report Dr. Shahid presented. So the UCSC team compared Kelvin's tumor to thousands of others and discovered that his rare sarcoma had molecular features like some adult lung cancers. This analysis helped his doctors to identify a treatment that they credit with extending his life about 2 years. So this was only a nominal success for this patient, but Now this portal has over 11,000 tumor transcriptome data, and it's open to researchers and clinicians worldwide, and likely helping other patients. RNA-seq technology has been undergoing rapid evolution as far as sample preparation, biochemistry, sequencing instruments, and bioinformatics pipelines. And there's a lot of room for improvement, but as is, it's already implemented as a clinical test by many institutions. So I was recently attended American Society of Human Genetics conference only a couple of weeks ago, and had a chance to hear clinical utility of RNA-seq by many institutions. For example, the first talk in session 63, the UCLA team discussed diagnostic utility of transcriptome sequencing for rare Mendelian diseases, where they studied 100 subjects participating to undiagnosed disease network at UCLA, and they performed whole genome sequencing along with RNA-seq. So what they shared was whole genome sequencing was sufficient for diagnosis of 31 patients, but RNA-seq, integration of RNA-seq with whole genome sequencing helped diagnosis of 7 additional patients. At this point, I would like to invite Dr. Moataz Ellithi to continue on clinical utility of transcriptome profiling from his perspective. Thank you so much.
Good afternoon, everyone. I'm a second-year internal medicine resident at USD here. Thanks a lot, Dr. Dogramaci and Dr. Shahid for the nice presentation. I wish mine is as good. So I know we just have like 10 minutes. We'll start off by kind of a glimpse about the power and versatilities of RNA-seq in different clinical settings, from screening to diagnosis to treatment options, and finally to prognosis and outcome prediction. So, as we all know, we have inherited the same recipe for life, which we call a genome. We can read the 3 million base pair of that code, but we can only understand 1 or 2% of what it does exactly. The rest is non-coding regions, which we used to call trunk DNA in the past, but not anymore. So, by using the RNA-seq data, this could help us to know about the downstream effect of those non-coding region variants, and if they are involved in the disease process and behavior or not. This will also help the scientific and clinical communities to further reprioritize the variants of uncertain significance, which we often detect on different DNA sequencing techniques. And to understand the value of this on the clinical side, imagine how enormous the data we can get from just one sample sequencing or whole genome sequencing. We often end up with few variants, or at least dozens of variants we don't know anything about. We call it variant of uncertain significance, just for the lack of better word. Probably we'll know more about their significance as we go. And as we move forward with knowledge, we know more and more about how impactful those alterations are in the disease development. And on the other side of this could help also scientists to focus more on the likely pathogenic variants, so they can focus the work more on those that will help expedite our knowledge. Here, example for that is this diagnostic pipeline done by Gunuraski and his colleagues in 2017. So what they did is they wanted to further diagnose patients with neurogenetic diseases that who were tested negative on DNA platforms. So they used different platforms, including DNA and RNA-seq, and they used data analysis pipelines to try to come up with a diagnosis for those patients, and they were successful in finding about 36% of patients having novel splicing, alternative splicing events. And this is just another figure to illustrate different types of alternative splicing and different possibilities. Now let's move forward to therapeutics. As we all know, we have here the COMPASS project, when we try to find out, sequence tumor and find out if we have any targetable genetic alterations. And we have had quite a success so far. Also, with the evolving paradigm of precision oncogenomics, scientists could be able, using RNA-seq data, could be able to design what we call tumor vaccines. And then the tumor infiltrating lymphocytes could be detected with RNA-seq through the same data analysis by detecting the human leukocytic antigen expression profile. This could give us an idea about the density of tumor infiltrating lymphocytes, and this has been correlated with response to treatment like immunotherapy. Now, even after matching patients with treatment, we still would be able to use RNA in prognosis and prediction response. I have here two remarkable examples, two studies. The first one done by Ehrlich and his colleagues. They used peripheral blood cell-free DNA clearance as a marker for response to chemotherapy. They found it as a good predictor for response to chemotherapy, and this also on the other side could open the door for those who haven't cleared their DNA for early consideration of alternative or additional therapy. This second one is a study done by a group from Sweden. They used the Swedish breast cancer genome data to come up with a multi-gene RNA sequence-based multi-gene classifiers as biomarker for disease. As we have been using for decades, these 5 conventional biomarkers are used to determine patients' prognosis and dictate pretty much of the adjuvant treatment. Hormone receptor status, estrogen, progesterone, human epidermal growth factor receptor status, HER2, the Ki-67 proliferation index, and Nottingham grade. So what they did is they tested this trained multi-gene classifier and compared it with conventional method. They had about 3,000 patients tested prospectively for about median follow-up of 52 months. What they found was comparable prediction of the outcome, except for those group of patients when they found that even additional benefit in using RNA-seq based data. As you can see here, all these patients tested positive. The yellow ones tested positive for hormone responsiveness based only on the histochemistry, but not on the DNA data. The blue one was concordantly positive on both. So those, as we would expect, had a better response to hormonal treatment and had better outcome overall. The yellow one, however, didn't show this similar or equivalent survival, and didn't show much of a response to hormonal treatment, but it did show significant response to chemotherapy. Speculating from this that probably it should be able to aid at least those subset of patients. Challenges: any power and versatile technology like RNA-seq will have its own shortcomings as it's developed. So I would like to have the clinical side of things here. First, due to significant inter and intra-tumor heterogeneity, as we all know, the cancer cells do not follow any rules. As they grow up and divide, they pick up more mutations. And I think clinicians as well as scientists should keep that always in mind keep in mind the difference in subclonal architecture, so that you will focus their treatment for curative intent on more suspected to be driver mutations. Lack of treatment options that target loss of function alteration, and this has been a challenge, especially in oncology. As we all know, P53 is involved in almost 50% of cancer development, and at this point we're still falling short in providing a treatment option for that. Lack of strong evidence in rare cancer types, and again, if you have a rare disease, probably you will have less funding, less clinical trials, and you will end up having difficulty applying technologies like this. Cost and insurance coverage. This is pretty much, I think, applicable to any new or expensive technology. Hopefully with time the cost will come down, but it can come prohibitive to some patients right now, but we are hopeful that as there's more data and more advances in the data analysis pipelines, it should come down in cost. Availability is a bit related to this. So even though some institutions are actively establishing unified protocols to use RNA-seq more readily, like here at Imagenalytics, the problem is applying or adopting that to the clinical, directly to the clinical practice is still challenging. And for many reasons, adoption means a continuum of things. First, availability, then needs, then cost, then trade-off between risk and benefits. And we're still quite falling short just because the diagnostic pipelines and the analysis pipelines are continuously evolving so far. I would like to end this talk with two non-medical journal articles. One on the left from Forbes magazine in 2011. The one on the right side is from New York Times in 2012, not too long ago. So the first one is about a 40-year-old toddler. His name was Nicholas Volker. He was diagnosed with a severe inflammatory bowel condition that was treated with every treatment available for IBD. The only problem is he never got cured better. He received multiple surgeries, including colectomy at the age of 4. So at that time, his physicians or pediatricians at University College of Wisconsin did something that seems strange at that time and unproven. They sequenced his DNA just because they were out of options. Fortunately enough, they found a mutation in a gene called X-linked inhibitor of apoptosis, which is pretty much unleashing the immune system, making it attack the body more. And this finding had led him to receive a life-saving bone marrow transplant, and he's still living up to this day.
And that's a non-cancer example.
That's
I love that you
yeah.
gave us an IBD gastroenterology.
Yeah, that was also very impressive. This one is a cancer example.
Yeah, sure.
So the more intriguing about this is it's not too long ago after this one. It's about a medical student back in the day, also from the Midwest, was the University of Washington, St. Louis. He was studying and aiming to be a physician scientist in oncology, but he never expected to be a research subject himself. He was diagnosed with ALL in 2003 while he's final year medical student. Received chemotherapy, but failed, relapsed. Received a allogeneic stem cell transplant, also failed, and had a recurrence almost in 2011 when he was starting his career as physician scientist in oncology. So his research colleague Timothy Lee decided to involve him in one of the studies they were doing at AML at that time. So they sequenced his DNA and RNA. Unlike the case for Nicholas, they couldn't able to find any targetable mutations in his DNA. But based on the RNA analysis, they found that one gene was overexpressed. This gene is called FLT3, which is a growth factor involved in lymphocyte proliferation. and this led him to receive sunitinib, which is a multi-tyrosine kinase inhibitor that inhibits this pathway, and he's still living now without any recurrence. I will just end up with a quote from the same article, and I think we all, as scientists and healthcare worker, agree with that aim. Thank you, everyone. Thanks for listening. I would like to thank Dr. Wilkie, Dr. Lugo for giving us this opportunity, and we will be happy to answer any questions.
Maybe one time short.
Any questions?
Cost.
We have our
How much does RNA-seq cost?
I
No
need
one's
to slow
gonna
down,
ask.
but I'm not up to date as far as cost. Cost may be affected by many things, the sequencing, really the bioinformatics pipelines include what kind of analysis
BGI
is
films
needed.
at $99.
How much?
I think $99 at BGI.
Just the transcriptional
But usually
profiling, or
some-
all the way through to the informatics
No,
final
no
product?
analysis. No,
Just the sequencing?
No
Yeah.
decision support.
But usually you're looking at
And how many
5 to
million
700
reads they
dollars.
provide?
I don't know the specifics. I
Okay.
guess 25, yeah, something like that.
25 million?
But 25 to 50 as far as I know, but I don't know the specific. I just know that.
Okay. So whole genome sequencing cost is around $1,000.
All the way through the IT pipeline.
I'm not sure. Adam, might you know any, Blake?
That's usually just for the sequencing itself.
That's
Gotcha.
getting the raw reads.
You can
essentially.
usually, yeah.
So
One
this
run
is
on the
cheaper
node
than
is
WCS.
$6,000, and you can get about 36 patients on there. So it's
Gotcha.
depending on the depth you want.
You want to wrap it up there, or you want more?
Thank you all again.
Thank
for-
you.
Good stuff. Moataz, who's the PI on Compass? Is that Steve Powell? You mentioned Compass.
No, Compass, I think it's Paul from the